My name is Jules Griffin, and I'm one of the group leaders at the Imperial College London Dementia Research Institute. And my group is interested in two interacting areas of research. So the first is looking at fat metabolism and how that relates to your relative risk of developing dementia. So we know that fat metabolism is linked to developing obesity, but also diseases such as cardiovascular disease and type 2 diabetes. It also turns out that fat metabolism is intricately linked to the development of a number of areas of dementia. And we've been looking at some of the mechanisms associated with Alzheimer's disease. We're also interested in looking at some of the microbes which help us break down our food in the gut, so termed the gut microbiome. So at the same time they help us digest our food, they also release important nutrients and small molecules which act as a signaling mechanism across the body. So we've been looking at some of the mechanisms associated with those. Now, in order to do this, we need analytical techniques that can profile samples from individuals at the epidemiology scale, so analysing thousands of samples. Typically, we'll be looking at biofluids, urine and blood plasma, and also we've looked at cell culture and indeed in some cases brain tissue. I thought it'd be useful though to show you what it looks like in the lab at the moment. So we're still under coming out of lockdown at the moment, so still under restrictions. And what you can see is that we've had to limit the number of researchers into particular areas. So I don't know if you can catch the sign in the background saying the maximum of 2 people are allowed into this area. So this area is where we have our NMR spectrometers. If I can just sweep around, there are 4 NMR spectrometers here, and I'll catch one in the background over here. This is what we use for looking at a lot of our urine samples and blood plasma samples to build a profile of what's happening in terms of your metabolism, and then we can relate it back to relative disease risk. I'm going to go over into a slightly noisier part of the lab over here to give you a feel for what the mass spectrometers look like. Different analytical technique, same idea here is that again we're going to be measuring small molecules called metabolites and relate that back to disease risk. What you'll notice is it's all quite empty at the moment. We're still under these social distancing restrictions, but hopefully very soon we'll have the lab back up and running and understanding mechanisms of why people develop dementia. Thanks very much for watching, and talk to you soon.